Welcome, and thank you for joining us today. My name is Ellen Wilson, and I'll be the moderator for this media briefing, which is hosted by the Johns Hopkins Bloomberg School of Public Health. Today's speakers will discuss mRNA vaccines, how they work, their effectiveness and safety, and their role in responding to infectious disease threats. I'd like to briefly introduce our speakers. Andrew Pekosh is a professor and vice chair of the W. Harry Feinstein Department of Molecular Microbiology and Immunology at the Johns Hopkins Bloomberg School of Public Health. He is also co-director of the Johns Hopkins Center of Excellence in Influenza Research and Response. Gigi Gronvall is a professor in the Department of Environmental Health and Engineering at the Johns Hopkins Bloomberg School of Public Health. We will have time for questions during this discussion. We will take some that have been submitted in advance of the briefing and some from the Zoom chat. If you have a question, please enter it in the Zoom chat addressed to panelists and hosts. Please enter your name, media outlet, and question. We hope to cover as many as possible. Please note that participants are welcome to use images, video, or quotes directly from the briefing, and that the content is for immediate release. Participants will be muted during this briefing and it will be recorded. Let's go ahead and start. Professor Pekosh, how do mRNA vaccines differ from traditional vaccines?
Well, let's start by saying that all vaccines basically do the same thing. They show your body something that is foreign, and they do it in a way that allows your immune system to recognize it and eliminate it. So vaccines usually come in two sort of flavors, what we call replicating and non-replicating. The non-replicating vaccines are when you give a person a known amount of a foreign protein and you want them to generate a response to that. Think about your flu vaccine that you normally get. The other type of vaccine is a replicating vaccine. That's where you get either a weakened form of a virus or you get something that is going to make the protein within the cells of your body. And the mRNA vaccines fall into that second category. They introduce a molecule called an mRNA that is coding this foreign protein. And it's actually the cells in your own body that make that foreign protein, and that starts to stimulate the immune system to recognize it. And it's that ability to express that foreign protein inside your cells that oftentimes makes mRNA vaccine technology so effective. It almost shows your body that an infection without having the really dangerous parts of a virus, for instance, as part of the process of generating that immune response. The most important part of mRNA technology, I think, is how fast you can make a vaccine once you know what you want to target. It's sort of like switching from a horse and buggy to a Corvette for travel. The mRNA technology still does the same basic thing, just a lot faster. Now, in that comparison, a lot of technological advances have gone into making that car, and it works very differently. But again, it basically still does the same thing as the horse and buggy, just in a much faster way, and in a way that most people would say is actually better.
Thank you, Professor Pekosh. Now a question for Professor Gronvall. Why are scientists excited about mRNA vaccines for diseases like influenza?
Great. Yes, thank you. mRNA vaccines can make flu vaccines more effective than currently and better matched against the viruses that we're all likely to be exposed to in the flu season. So every year, the flu shot needs to be reformulated. That's why you have to get a new shot every year, because it depends on what flu viruses are circulating and how much they've evolved from the previous year. So every year, experts from the World Health Organization, the CDC, other health agencies around the world, they meet and they make predictions on what vaccine needs to protect, what viruses the vaccine needs to protect against, and what things are. They look at the surveillance data to see what is going to be the likely problem in flu season. Because the manufacturing process is so long, especially for vaccines that are manufactured in eggs, they have to predict which flu viruses are going to strike at flu season many months in advance. So they meet typically in late February to start a process that ends with having flu vaccines ready in August or September in time for flu season. Sometimes, based on the data that they see, the prediction that they make is quite accurate, sometimes less so. But mRNA vaccines don't need to take as long to manufacture as traditional vaccines. So instead of 5 to 6 months to make, they can take less than 2 months to manufacture. So that means that they can start making them later, closer to flu season, which gives more decision flexibility. So which flu viruses should the vaccines protect against? And it can also, so that whole process can take months later. This should lead to vaccines that are better matches for what flu viruses people are actually likely to encounter during the season. So even in those years that there is a bit of a mismatch between the flu virus and what the flu vaccine protects against, I want to be very clear that everyone should get a flu shot every year. Flu vaccines are really good at reducing hospitalizations as well as deaths. But with mRNA, the hope is that it will be even more effective at reducing sickness, hospitalizations, and deaths, and making the flu vaccines perceived as better. Just, people notice that they're better will help with uptake. So again, mRNA vaccines can make flu vaccines more effective and better matched against the viruses that you're likely to be exposed to during the flu season.
Thank you, Professor Grunwald. Now we will take additional questions. And a reminder, if you have a question, please enter into the Zoom chat with your name, media outlet, question, and to whom you'd like to address your question. Let's start with Richard Payerchin with MJH Life Sciences. For Professor Grunwald, what are the best ways for primary care physicians to explain this complicated medicine to patients who may be skeptical or fearful about mRNA vaccines?
Sure. So there were lots of rumors and false information floating around during the worst parts of the COVID-19 pandemic. And some of those falsehoods have unfortunately remained stuck in people's minds. So it's really worth going over a few of the things to set the record straight. mRNA vaccines do not hang around in your body for long. They do not get incorporated into your DNA. While they were new to most people who didn't hear about the concept of an mRNA vaccine until COVID, they'd actually been in development for years. Cataline Carrico and Drew Weissman from University of Pennsylvania got the Nobel Prize for their foundational work for mRNA vaccines. And it's important, their work was published in 2005. So it was in process for years before then. So they've been around for a while. mRNA vaccines also do not cause turbo cancer or regular cancer. They have no effect on fertility. And importantly, they saved so many lives. The first year, it's estimated they saved over 14 million people worldwide and prevented many more hospitalizations. And that number, the hospitalization number is really big. It's much bigger than the 14 million people. And people should remember that you don't just bounce right back after being in the hospital. Being intubated, it's a very disorienting experience. It's not fun at all. And so these vaccines save people so much pain and suffering, not to mention economic loss.
Great, thank you. Here's a question from Meg Winggerter with the Denver Post for Professor Pekash. Does mRNA technology have enough momentum to keep moving forward without federal support, especially given that the FDA seems less friendly toward products that use it than it was before?
Yeah, I do think that there's enough momentum to keep mRNA technology development going. Many other countries are supportive of mRNA vaccines, especially for infectious diseases. Progress will be slowed by less U.S. support because the NIH just provides so much basic science research support that could help move these technologies forward. But the work on vaccines will continue. The FDA has noted that they will continue to fund mRNA vaccines for cancer treatment, so that's a good sign. But I think overall it will continue to progress, though at a slower rate without a greater emphasis from NIH on their utilities, particularly for infectious diseases.
You mentioned cancer. Here's a question from Gabriel Etzel with the Washington Examiner for Professor Pekash. Can you explain how mRNA vaccines work for cancer? Do they work in a preventative or treatment capacity? How far away are we from using this technology to treat the most common forms of cancer, like breast or colorectal cancers?
Yeah, right now, the mRNA vaccines that are targeting cancer are what we call therapeutic vaccines. So once you develop the cancer, you can then design a vaccine that targets some of the unique proteins and other things that those cancer cells are showing to your body. And therefore, your immune system can get ramped up and target those very specific cancer antigens that the cells are showing. Um, there are some cancers that have very common antigens, but there's also an interesting area of research about personalized vaccines, which may mean that that a physician could go in, test your cancer, find some of the unique proteins that those cancers are showing, and then design for you a personalized messenger RNA vaccine that can target your specific cancer. Again, much of this technology is in its early stages of development, but it's really, really fascinating to think that one could really design this kind of personalized medicine relatively quickly, and then hopefully have that increase the efficacy on which these treatments can work on an individual basis.
Can I
Great,
just add
thank.
to that?
Oh, yes, please go
So
ahead.
there are two vaccines, perhaps more, but two one that you know of that do prevent cancer, and that is the hepatitis B vaccine that's given to babies, and the HPV vaccine, which is given to early to tweens and teenage years. And so there is every reason to think that mRNA will be used as a platform for those for both of those cancers, to prevent those cancers and possibly others. There is a lot of research about different infectious disease causes for a variety of cancers and other conditions that were not thought to be connected to infectious diseases early in life. And so, as we learn more, this is a great platform to be applied to it. So I think the future there is really bright.
Thank you so much. Here's a question from Apollonia Adeyemi with New Telegraph for Professor Pekash. What makes mRNA vaccines different from others, especially in terms of potency?
I think there's a couple things. As we talked about earlier, the speed in which you can make them is really sort of unparalleled. All you really need is the sequence of a target, and you can then, within days, make the pilot mRNA vaccine that you can test, and then relatively quickly scale up to generate larger amounts of it. I think the second thing that's interesting about them is they oftentimes do show a high level of potency, although again, some of this is dependent very much on the target and what that target is, and the importance of antibodies, which are the primary things that mRNAs have been good at generating, how good those antibodies are at protecting. Um, as I mentioned before, what's interesting about mRNA technology is that it introduces this mRNA into your cells. Your cells make this foreign protein, and your immune system sees that cells as if it was a virus infected cell, as something foreign. And therefore, much more of your immune response gets initiated in a much more faster and stronger way. So, as opposed to things like your flu shot, for instance, which again is good, but it isn't able to generate as strong and as broad of an immune response as an mRNA vaccine because of the way the mRNA vaccine technology shows that foreign protein to your immune system.
I'd like to add to that with every vaccine that gets approved by the FDA, it has its own process. So this is, even if it's a platform technology that the FDA is familiar with, and the review process may be expedited in the case of the flu shots that you get every year. There is, every vaccine is considered, generally separately. And so if our mRNA is being applied to one disease and then another, they are going to have separate technical considerations for each.
Great, thank you so much. Professor Pekash, could mRNA technology help make the flu vaccine more effective?
Absolutely. I think Dr. Gronwald mentioned the speed in which mRNA vaccines can be made. And what that means is that would give us the ability to wait longer into the flu season and make a better choice about what particular virus we want to make a vaccine against. The other way that's, again, specific to influenza vaccines is that most of the influenza vaccine is grown in eggs before that's processed to generate the vaccine. That process of growing influenza in eggs can sometimes introduce mutations that make those vaccines less effective at inducing responses to the current circulating vaccines. And mRNA technology would avoid those types of mutations completely because it would be based completely on the authentic sequence of the virus that we think is going to be dominant in next year's flu season. So it's fast and it avoids some of the complications of egg-grown influenza vaccines that both contribute to reduced efficacy of the current flu vaccine.
Great, thank you so much. Professor Grunwald, mRNA vaccines became widely known during COVID-19, but what do you see mRNA technology being most useful for?
Well, we already talked about how mRNA has a really bright future in cancer therapeutics, and the Moderna CEO signaled that that would be the basis, at least for that company, for the American market. But I want to emphasize that mRNA vaccines are important for pandemic preparedness because of their effectiveness and speed. Specifically, canceling the contract for the mRNA vaccines for H5N1 avian influenza was a strategic mistake. There is no one who could say if or when H5N1 bird flu will become contagious between people, but it is a risk that is made more serious because H5N1 is endemic in the United States. It's already causing lots and lots of problems for the agricultural sector. We have reduced public health surveillance and a lot less interest in performing surveillance. Other traditional vaccine approaches, like Andy just said, Dr. Pirkos just said, use eggs, which is a slower technology, but it also presents some technical challenges in making the vaccine for H5N1, which is highly lethal in birds. The NIH director, Jay Bhattacharya, has said that people do not, quote unquote, trust the technology, the mRNA technology, and that is why they are investing in older, less efficient technologies. But his job is to create trust in the proven technologies that will help save lives, and giving Americans a less effective product will not help to do that, especially if people see that mRNA vaccines that are more effective are available elsewhere in the world.
Thank you so much. Here's a question from Rachel Nania with AARP Magazine for you, Professor Grunwald. What is the likelihood that we will have an mRNA flu vaccine for the 2026-2027 flu season?
So, as you know, the mRNA vaccine that was submitted for regulatory approval, there was some drama over whether or not it was going to get reviewed. It's impossible for me to predict what the timeline will be for the review and what the outcome will be. And it should be noted that FDA is working with a much slimmer staff, and I don't know how that will affect the turnaround time. But these are and other questions are really best placed to the government officials at HHS, especially the secretary, and whoever is replacing Dr. Vinay Prasad.
Great, thank you so much. Professor Pekash, what are some of the side effects of mRNA vaccines?
Well, side effects really are driven by two things. One is the platform itself. So as many people talk about, the mRNA vaccines do generate a strong, robust immune response. What comes with that is those feelings of aches and pains, redness at the site of inoculation. Again, with COVID mRNA vaccines, a lot of people did talk about being tired for a day or two afterwards. Those kind of non-specific symptoms are probably the most common ones. Sometimes there are some very specific symptoms that are resulting from what the vaccine actually has. And in some cases, the incidence of myocarditis in some young males is something that sort of speaks to that specific part of the response, where a very small percentage of young males who were getting the COVID vaccine ended up with this myocarditis, which is an inflammation of the heart. It's important to say that the rate of myocarditis from COVID-19 infection was significantly higher than that from vaccination. So even with those side effects, the vaccines give you lower side effects than the actual infection. And I guess the last thing I'd say is we talk about mRNA technology, and I want to make sure people understand that this is not a static technology or a static platform. Many companies have now second or third generations of these mRNA vaccine platforms that actually have been designed to reduce some of those side effects and to make them a little bit more acceptable in the population. So this technology is not frozen, and it is constantly evolving. And some of the concerns that people voice about wanting something with less side effects or with more efficacy are actively being investigated by both academic laboratories as well as by companies.
Can I
Great,
just add to that, that
please.
there's often a perception that if people don't have side effects, that it's not working, and so just the reverse of that concern, and that is also misplaced. This is not, in general, no gain, no pain, no gain approach does not apply to the human immune system. So that some people will have side effects and some people will don't. The vaccine companies are working to reduce the side effects, but people should not be concerned if they get this vaccine or any other vaccine that has known potential for side effects, like for shingles, that if they don't get side effects, that's not working. That's not the case.
Great, thank you so much. For Professor Prakash, a question from Lena Sun with the Washington Post. There is an increase in a COVID-19 variant now that seems to have stronger immune escape. What do you see as the challenges now in the current anti-mRNA environment for the COVID vaccine manufacturers if this variant increases in virulence and transmissibility?
Well, some of the challenges with COVID-19 are very similar to the challenges that we have with influenza vaccines. Now that COVID is replicating in the human population, it's evolving in slightly different ways depending on geography and many other factors. So what we have is a number of different variants that emerge with COVID-19 viruses, very similar to what we have with influenza viruses. So right now, the preferred method for COVID-19 vaccine choice is to try to select one variant and that be the main component. This new BA 3.2 variant that's circulating is very different from all the other COVID variants that are circulating right now. So there's a difficult choice to be made between two very different variants for the vaccine. Potentially, there could be what we call a bivalent vaccine, meaning that it could contain two strains of COVID-19. And that may be something that the FDA and other regulatory agencies may be considering to allow the vaccine manufacturers to do. But we will expect an update on the COVID-19 vaccine formulation to happen in the next couple of months in preparation for continued rollout in the fall. Again, the FDA has loosened some of its recommendations, or I should say tightened its recommendations to very specific populations for the most part. But COVID vaccines will be available in the fall, and similar to influenza vaccines, there is a lot of evidence suggesting that people should go out and get those vaccines because they are safe and they are effective.
Great, thank you so much. Professor Gronval, we're seeing growing investment in mRNA technology around the world. How are different countries approaching this, and what are the implications for global health equity and future pandemic preparedness?
Sure. Yeah, it was clear to the rest of the world how many people were saved during COVID with this amazing technology, but we can't forget that it was very unequal. That's why international organizations like not only the WHO, but the Coalition for Epidemic Preparedness Innovations, or CEPI, exists to make sure that this vaccine technology is available for less resourced countries and not just the ones that can afford to protect their people. Because we know that in the future, there will not just be flu viruses that come around every year, but there will be future pandemics, and we do need the infrastructure in place to be able to protect against them. And infectious diseases very famously do not stick to borders. So we've had several pandemics within most of the people on the Zoom have lived experience with several pandemics, and we can expect that there will be more. So nobody can say when, but it's important to have that infrastructure to be ready for them in place.
Professor Pekash, looking ahead, what excites you most about the potential of mRNA technology for diseases like influenza, and what are the key scientific hurdles that still need to be addressed?
Yeah, I think the ability to make better, more efficacious vaccines than our current ones is probably the most exciting thing. Again, our current flu vaccines work, but they can be better. And mRNA technology represents a platform that could easily allow us to make great strides in improving influenza vaccines simply because we don't have to make the vaccine choices early, and we can be much more precise about the sequence we put in the vaccine and make it a better match. Those are two very low hurdles that we've not been able to really find a way around for dozens of years with flu vaccines. And then I think the ongoing research to, again, try to minimize side effects, to maximize the efficacy, to reduce the amount of the mRNA vaccine that we get that allows us to still generate a strong immune response are all really powerful ways that these vaccine platforms will get better over time and continue to improve. So I think those are the two things that really excite me about this technology.
Great, thank you so much. And the last question from Gabrielle Etzel with the Washington Examiner for Professor Gronval. We have talked about the role of the FDA in this, but how will the turmoil with the CDC's advisory committee on immunization practices affect vaccine rollout this fall?
Yes, it's impossible to say at the moment now that the ACIP was determined to not the actions were not determined to be legal, and there's recent drama now that we're in our post-Malone phase of ACIP. But all I can say is that I can't predict what's going to happen there. I would only say that the ACIP is a very important committee, and it's important to have expertise. You know, you need to have the expertise to know vaccines, to know children's vaccines, and we need to create an environment where the American Academy of Pediatrics and other experts are able to participate, and we can talk about the real risks, the real data that we have, because without it, we are not doing the best that we can to provide vaccines for the American people. And so I hope that we can get back to a place where we can rely on the scientific judgment of all of the advisory committees, that not just the ACIP, but all the rest of them within the Department of Health and Human Services.
Thank you both for answering all the questions. We will now take a few minutes to share brief closing remarks. Professor Pekash, please go ahead.
Yeah, I just want to emphasize that mRNA vaccine technology allows for that rapid generation of new candidate vaccines. It also allows for a very potent response to those different foreign proteins. And we need to continue to invest in that technology because it's quite clear from all the data that was generated with the COVID-19 mRNA vaccines, as well as from other mRNA vaccine clinical trials that have gone on since, that this technology has a great deal of promise. It also has areas where we can continue to improve it and make it better and safer. And without that investment in basic research on this technology, we won't be able to move forward and really understand the true benefits of this technology for infectious disease vaccines, as well as for other vaccines, such as for cancer.
Thank you, Professor Pekash. Professor Gronval, please go ahead.
Just that we should be investing in the best technologies that we have to save lives and keep working to make those technologies even better. That's the biological future we all want, where we can save lives. And it's important that all of the people involved in making and delivering on those technologies remember that.
Great. Thank you so much, Professors Pekash and Gronval, for your insightful answers. We will email the links to the resources mentioned today and share it in the chat. And with that, I'd like to say thank you again to everyone for joining us. A link to this recording and an unofficial transcript will be provided later today.